Here we go. So we'll look at the warm-up. And again, we'll be just building on these concepts that we started on Tuesday. Today, we're going to continue talking about transport across membranes using membrane proteins and just taking that up a notch and getting way deeper in like how exactly does a ligand-gated channel even work and what are the different kinds and things like that. Here we go. I need to share this screen. All right. So name at least two different ways ion channels in a cell's membrane can be gated. Here, most people who answered this kind of got this right. Again, we have voltage gated, which are going to be at the core of, action potentials and things like that once we get to signaling in the nervous system. There's ligand gated, which is how you have one neuron actually talk to the next neuron using synapses and neurotransmitters. Ligand-gated channels also how your taste buds work, right? You have a channel on your tongue that is operated by the presence of sugar. And if sugar is there, it opens a channel to let sodium in, which creates an electric current, which sends a message to your brain that there's sweet in your mouth. Mechanically gated, we talked about that's at the core of how your body knows your stomach is stretched out or you're being touched or things like that. Some people wrote in their answers, like mentioned carriers and things like that. And that's kind of going off into a different kind of transport protein, right? Channels, these gated channels, and not all channels are gated. Some channels are just always open. They're one flavor of transport protein. The carriers were another thing. So like it wouldn't make sense to mention carriers in your answer to this question. Just kind of, it seemed people were kind of getting a little unclear about carriers are one class and channels are another class. So just kind of clarifying that. Cell organelles, again, hopefully, the things that make protein are ribosomes. Some people wrote rough ER. I gave you credit for that. Rough ER, it's the ribosomes on the rough ER. That's actually the place where the proteins are being assembled. But, in general, that is a place where proteins are getting made. ATP, the mitochondria. I think depending on the time, we'll probably have a chance to start getting much deeper into what exactly is going on in the mitochondria today.
I have a quick question. I wrote mitochondria and I got it wrong. So I don't really understand. And I can email you a screenshot of it, but.
Right, you probably misspelled it or something like that, right? The system, I have to give it the possible answers. So I go back manually and I double check. Like if you wrote my mitochondria with spelled it off by a letter, I give you credit for it. But
Okay.
the machine isn't smart enough to like say, oh, Jean like typed an E when she meant an O or something. So that's something I just go back and manually do.
Great. Thank you.
So this one's a little trickier. And this is going to be, here we only got like 3/4 of the people who did answer got this. And we should talk about this because this will be important as we continue on. If a cell membrane has a voltage of -70, and again, we have to remember that convention is the voltage is the inside with respect to the outside. So if it's -70 millivolts, it means the inside is more negative than the outside. So that's important to remember. Whenever you see this voltage, it's the inside compared to the outside. So then so that means the inside is more negative and the outside is more positive. So then if I have a cation, like sodium, opposites attract. So which way does it want to go?
Inside.
It wants to go inside. It wants to go to where it's more negative. So this is something you're going to want to try to get comfortable with, because it's going to become more and more important over the coming week or two as we start talking about electrical properties of cells and signaling and graded and action potentials and all that. So this is something that's in parallel. Like, we're going to spend more time today talking about diffusion and about how sodium might want to move one way or another depending on concentration gradients, the chemical force based on how much sodium is on one side, how much sodium is on the other side. And sodium is going to want to move from higher to lower concentration by diffusion. But then another thing that is happening at the same time might be this electrical force. And it might be in the same direction or it might be in the opposite direction. So you have to think about it as a separate thing that's happening in parallel to the diffusion and chemical force that's moving things. So we will be getting much deeper into all of that. Choose all that are true about facilitated diffusion. All right, and this was another thing we're going to continue going deeper today. It is not a form of active transport, right? Things in facilitated diffusion just go along their concentration gradient. It has a name, like it sounds like help, like it needs energy, but it's not true. It does require special membrane proteins called carriers. In fact, that's the definition of it. It is going to be diffusion across a membrane that is facilitated by carriers. It is a form of passive transport. The stuff is going to move whichever direction the concentration gradient is making it move. The big thing that we talked about, remember I drew that picture where we saw regular diffusion versus facilitated diffusion. There's a limit to how fast stuff can move across. At some point, there's a maximum rate that things can move across using the carriers. So these three are the correct ones. It's not active. It is limited, so no limit is wrong. It doesn't require ATP, right? Because it's just being driven by the energy and the concentration gradient. In a few moments, we'll be looking at other membrane proteins that do pump things against their gradient using ATP, active transport. But this is not active transport, this is passive. Are there any questions about this? This is important to really understand facilitated diffusion, because we're going to continue developing related ideas and then bringing facilitated diffusion back in and seeing how it works together with the other types of transport.
So whenever we're talking about facilitated diffusion, there are carriers involved.
By definition.
Okay, thank you.
Yeah, by definition, that is what facilitated diffusion is. It's diffusion that is occurring with the use of carriers in the membrane.
So then just to be clear, like the ligand-gated channels, those are active transport, but is there any other?
Absolutely not. Those ligand-gated channels are still passive transport once they're open. They have a gatekeeper, but once they're open, whatever ion they are letting through is just going to move to one side or the other based on its concentration gradient. We're going to talk much more about active transport today. Active transport is when you are using the energy, usually from ATP or something else, and you're pushing things against their concentration gradient from low to high concentration, opposite the way they would naturally want to go due to diffusion. So gated channels are not active transport. Does that make it clearer?
Yeah, kind of. I think it'll make more sense as we talk more about it.
Okay. Choose all that true regarding cilia. So they can move everybody, well, everybody, the people who answered got that. They are definitely not important for endo and exocytosis. Yeah, the particular membrane protein we talked about that's important for that was like clathrin and the clathrin coated pits and stuff. Primary function is not to increase area. What is the primary function of, or which membrane modification is used if you want to increase surface area?
Microvilli.
The microvilli, exactly. And then they have the microtubules. We talked about that 9+2 arrangement of microtubules and how they have those little these little proteins in between that pole and make them bend and wiggle. So that's that. La la la, la la la. All right, so what we're gonna do now is continue talking about membrane proteins, and specifically we need to continue talking about the membrane proteins involved in transporting things across the membrane. So membrane proteins continued. And specifically, we're talking about ones that are involved in transport. So the things that can help things move across a membrane. We talked about channels. These can be gated. These are usually for ions. Right, so that was one thing we talked about. Remember, and then we said there's ones that'll let sodium through or potassium through or chloride through or some like group of different things. There were carriers. These are like, for sugars and amino acids. And these were the things that created the facilitated diffusion. So once these are open, things will still just move according to their concentration gradient. Maybe I should just do a little aside real quick about active versus passive transport, since it seems like that would be helpful. So passive transport, this no extra energy needed. Extra, read all about it. No extra energy needed. So basically, things move. It's going to be faster if I type this puppy. Things move along. Actually, things move along their concentration gradient from higher to lower concentration. For example, diffusion or facilitated diffusion. Osmosis, which is just a special kind of, these are all just happening using the basic energy that the molecules have. The fact that anything that's not at absolute zero is wiggling and jiggling. So active transport, on the other hand, I'll use a different color. This requires energy. So we're going to look at a variety of these in a few moments, in just another few moments. But things that like pump ions across the membrane against their concentration gradient using the power of ATP would be example of active transport. We'll see a couple of different versions. Some active transport is going to be powered directly by ATP. Some is going to be powered a little more roundabout. Again, we're going to be looking at primary, secondary active transport in just a few minutes. But just to give you the bigger picture, passive transport, you do not need extra energy. It's just based on the energy of, in fact, I can do another thing. Let's, diffusion. How does diffusion work? Again, diffusion works without any energy because things are wiggling and jiggling. Like, let's think about, there's a lot of stuff over on this side and not so much over here. Right, so you have to imagine, we're not in an equilibrium here. This is just a snapshot right at this moment. Here are a whole bunch of molecules, let's say they're sugars or whatever, on this side. And on this side, there's not many at all. And then remember, anything that's not at absolute zero is wiggling and jiggling and then zipping through space. So these little puppies are, it's like zip, zip. They're going at, all these different velocities, zipping around, because that's just what little molecules do. They're just moving around. Again, that's what temperature is. It's just the energy as they're all zipping and ziggling and moving around. And now imagine you are trying to go towards this area where it's higher. Remember, this is kind of the high concentration area. This is the low concentration area. If you're moving towards the low concentration area, you're going to be able to go a lot farther before you run into something and then hit there and change direction. If you are trying to go the other way, boom, you are going to hit something and end up shifting your direction pretty quickly, because you don't go very far before you whack into something if you try going this way. But if you're going this way, you go much farther before you run into something. So the net effect is there is a movement, a bulk flow from high to low concentration. But it's just based on just the fact that these things are moving randomly. These are all random. Call them like random walks. But the fact is, if you randomly are starting to walk towards the side of low concentration, you are probably going to go much farther before you run into something, right? The metaphor I use sometimes, if you took a bunch of kindergartners and you just fed them a whole bunch of kind of candy and had them drink a bunch of Red Bull and all this stuff, and they're just like, oh my God, blah, blah, blah. And then you roped them all into the corner of the room so they can't move. They're all stuck there in the corner of the room, just wanting to explode. And then you set them free. They're not thinking like, oh, there's a high concentration of us in the corner of the room and there's a lot of free space out on the other side of the room. Let's have a net movement from high to low concentration. They're just going to run and bounce off the walls or bounce off each other. And it's just going to happen that they're going to start flowing from high to low concentration of little hyperactive kindergartners. That's what diffusion is. These little molecules are just randomly bouncing off the walls and bouncing off each other. But because of just the nature of there just being more things blocking their path, if they go this way, the net movement is this way. So any questions about this? So this is your class of passive transport because you don't have to add any extra energy to have this net movement this way. Whatever energy they got just from existing is driving this process. If we wanted to make them go the other way, then we would have to fight that, and then we would have to add the extra energy in with active transport. So diffusion, again, diffusion is going to be at the core of our lab today. It's going to be at the core of our lab on Tuesday. It's at the core of our discussions about physiological processes for the next 4 months. So you really want to kind of have an intuition about diffusion. All right, so that's kind of that, looking at this passive transport in more detail. And again, facilitated diffusion is the same thing as diffusion. It's just, it has, the molecules need to use a carrier to move from one side to the other side, which results in this rate limit of how the ultimate speed or rate that things can move from one side to the other, but they're still going to move along their concentration gradient from high to low concentration. So there's no extra energy. The only difference between facilitated diffusion and regular diffusion is that there's a rate limiting quality to it. And osmosis is just going to be the specific example of diffusion of water across a semi-permeable membrane. Rather than, normally we think of some solute diffusing through water, but in osmosis, the solute is trapped. Semi-permeable membrane. The solute can't move. Normally, which way, assuming there was no membrane, let's give these, this is side A and this is side B. If there was no membrane there, which way would the solute want to move?
To side B.
Exactly. It would move along its concentration gradient from A to B. So if there was no semi-permeable membrane, the solute would just move by diffusion from side A to side B. But I have created this barrier here, this semi-permeable membrane, which blocks it. So it just stays high concentration on side A. However, semi-permeable means water can move back and forth. And what this creates is a situation, it's kind of an awkward way to think about it, but you could say if this is just distilled water here, that's the highest, if you're thinking about concentration of water, which is kind of a weird way to think about it, this is pure water. It's the highest possible concentration of water you can have. This side here, the water is kind of diluted with solute. There's less water, on this side. So you could say this is higher concentration of water, less concentration of water because there's other stuff there. So water is going to move on its concentration gradient. The water is going to want to move from kind of the higher to the lower concentration of water. So that's osmosis. I have to say, though, it's much more intuitive for me to think about the saltier side kind of drawing in the water. For me, again, maybe it's because when I was a kid, I did the kind of, unfortunate thing of, sprinkling salt on a slug and watching the poor little guy like shrivel up and realizing, oh, salt draws water out of things. But, you can think about whatever side is a higher osmolarity is going to basically be pulling the water. The water is going to be drawn towards the side of higher osmolarity. So whenever you're trying to figure out which way does the water move in osmosis, it goes to the side that's saltier, the side that has higher osmolarity. So does that make sense? So again, we'll be looking at this in our lab today. We're going to be looking at our lab on Thursday, next Tuesday. And again, we're going to be looking at this for the rest of the semester. It's just all the time we're talking about osmosis. And again, for me, the intuitive way to think about it, whichever side, if I have some membrane here and one side is more stuff dissolved, one side is less dissolved, water always goes towards the side that has more stuff dissolved in it. If this was 3 osmolar and this was 2 osmolar, the water is going to move from the lower osmolarity to the higher osmolarity. So you always go, the side that has a higher osmolarity is always the side where the water is going to move. Right. This is why you can preserve things by salting them or candying them, right? If I have some little piece of, some fruit, some mango or something, and I, add sugar or salt to it, or, then the water gets pulled out of it. Any little organisms that are wanting to grow there are shriveling up and dying. So if you add sugar and salt to things, you basically are desiccating any little bacteria or something that is trying to grow on there. So it's not going to, make this thing go bad. So, that's why, yeah. So preserving things by either salting them or candying them basically is creating this really high osmolarity, which then is going to take whatever little microbe lands on there and turn it into a raisin. So, all right, back to our membrane proteins. All right, so again, these asides are useful again for the rest of the semester for our lab, and as we continue looking at these transport proteins, because we're going to see, so far we've just kind of looked at passive transport, but now we're going to start getting into kind of more complicated transport proteins that do more kind of more fancy things. So some carriers are just kind of simple, like revolving doors. So the ones we've looked at so far are just going to let like glucose move in or out depending on its concentration gradient. or an amino acid, for instance, move in or out depending on its concentration gradient. I'm going to talk about now are co-transporters. So co-transporters they're kind of more fancy versions of these carriers that couple the movement of more than one molecule across the membrane. And these can be antiports or symports. So let's say here's my membrane. An antiport would be something that allows, for instance, allows potassium to come in, but only if sodium leaves at the same time. You know, a symport would be something where they have to come in together, like, you know, glucose can come in, but only if sodium comes in at the exact same time. So there's like these docking stations, like there's kind of like a dock for glucose, there's a dock for sodium, and this revolving door will only run assuming that both of them are sitting good, ready to go. And once they're both there, then the revolving door will spin around and spit them out on the other side. But this couples co-transporters, these coupled transporters, right? It has to, like this one here, potassium will cross, but only if there's a sodium that is getting pushed out at the exact same time. So this is another class of transporters that you need to know about. And we're going to see how these actually work for some fancy secondary active transport soon. So does this make sense? Another thing I should mention, you know, there are pumps. Whoops. Like for instance, something that pumps sodium, but let's say there's already a lot of sodium out here and not much sodium in here. Sodium does not want to go out because there's already a lot. In fact, by diffusion, the sodium wants to come back in. So if I want to keep pushing the sodium out of the cell up to where there's already a bunch, I'm going to need energy. And one way I can do it is with ATP. This is what I call primary active transport. Primary active transport is directly using the power of ATP to pump things against their concentration gradient. So does that make sense? We'll see a variety of these things. We'll have pumps that pump ions or pumps that pump different things. Actually, I should mention, if I just have my kind of classic cell, one of the pumps that we're going to see is that something that pumps sodium out and pumps potassium in. And it's going to use ATP. So this sodium potassium pump, this is going to be happening in a lot of your cells, that all of our neural cells and all of our muscle cells and stuff. So one of the things we're going to talk about, the intracellular fluid, the cell inside a cell, you usually have very high concentrations of potassium. Outside the cell, you have very high concentrations of sodium. And that's happening because these pumps are constantly just pumping sodium out of the cell and pumping potassium into the cell. This is going to be really important as we go on. In fact, I can just intracellular fluid is going to be very high in potassium. Extracellular fluid is going to be very high in sodium. You know, this is going to be important because then if we're just thinking about diffusion, if I open a sodium channel, which way does the sodium want to come?
Inside the cell.
It wants to come into the cell because there's a lot of sodium outside, not much inside. So sodium wants to move in, potassium wants to move out if we just look at diffusion. There's also more chloride outside, more calcium outside. So this will be important when we kind of look at which way do things want to move when we open up channels. You know, it's going to matter knowing which stuff is more outside versus inside. You know, and this, again, is being maintained because we're pumping sodium out, pumping potassium in. So we're doing this actively. We're creating this and maintaining this difference actively using primary active transport. You know, so we'll be coming back to this.
Would you mind going back to the definition that you had for the primary active transport? I just didn't finish writing it down.
It's just using the energy of ATP directly to pump things against their concentration gradient. Like in this example, so there's already a lot of sodium outside. Sodium does not want to go outside because of, you know, the diffusion gradient is pulling it back in. So I have my special little pump here, which is coupled to breaking down ATP using this energy to push it against its concentration gradient.
Thank you.
Right, it would be like if I had a, you know, a fountain. I don't know, some fountain. You know, if there's water, and there's like, let's say there's a hose that goes up the middle of this thing. if I just let it sit the way it is, the water is just going to stay there. The water is not going to do anything. If I actually, because gravity is pulling water down. If I actually want this fountain to work, I got to put a pump in there and I got to plug it into like some power source. And then that little pump with its little propeller will, and now I've got water fighting against gravity and making a nice little fountain. But I can only push water against gravity with some energy source that is driving that, that active pushing against gravity. In this case, I'm plugging my pump into the wall and getting the electricity. But in the case of, active transport, you're getting the energy from breaking down ATP instead of plugging these little pumps into the wall. So that make it clearer?
Yes.
All right.
I have a quick question, actually. Sorry. So just to clarify, so co-transporters are just basically like passive because it just requires like for example, antiports require like a potassium and a sodium in order to move. But then so what's the driving force? Is that still technically passive transport?
Yeah, so that's a good question. So it gets messy. It gets messy because, for instance, where did I just put it? This is kind of an antiport that's also a pump. So sometimes they get coupled into primary active transport like this. Also, as we're going to see in just a few moments, secondary active transport is going to use the so this thing that I drew here, it is basically passive on its own, but it actually is going to get kind of blurry about passive versus active when you put it in a larger context, which I am going to explain right now. So yeah, your question it's a good question, and the answer is messy. So here we go. So primary active transport. Use energy of ATP directly to pump things against their concentration gradient. There is another kind of active transport called secondary active transport. So let's look at that right now. So here, active transport. Let me just use my typing tool because it will make things quicker. So this is it sounds weird. It's gonna make way more sense once I just show you how it works. So the way it works is like this. So here's a cell. And let's say here's glucose out here. There's already a lot of glucose in the cell. So what I do is I have one of these coporters, a symport, that moves glucose in, but only if it comes in with sodium. And then I put a sodium pump here. And this is going to be primary active transport. So down here, let's do a little spotlight. Let me draw that better. So primary active, I'm using the power of ATP to pump the sodium out, which means it's low sodium, high sodium outside, low sodium inside. So sodium wants to come in. So this is creates a sodium concentration gradient. Right, and if you remember the very first week of class, we talked about one of the types of potential energy that is going to be important in our class is the energy stored in a concentration gradient. So this actually kind of stores potential energy. Right, it'd be like if you pumped a whole bunch of water up the hill to a reservoir up the mountain, and then if you let the water fall back down, you can use it to run the paddle wheel and mill your grain or something, right? If we're pumping a lot of sodium out, so there's a lot of sodium outside compared to the inside, the sodium wants to come back in along its concentration gradient. And that's where this little symport is going to come into play here. So I've got my little symport here, which couples the movement of glucose with the movement of sodium. So this is kind of getting back, I think it was Courtney's question about, is this passive or active? Things are going to move along their concentration gradient, but we've created a situation where the sodium really wants to come back in. And sodium can only come back in if glucose is docked alongside it. So the sodium is like, oh, want to go back in. Oh, glucose, God, boof, glucose. So this basically ends up pushing it starts pulling the glucose in along with the sodium. The sodium is going to come in because there is this big gradient, but it can only come in if it drags glucoses along in with it. So what we are actually doing is, this is called secondary active transport. Secondary active transport, using the energy stored in the concentration gradient of some other molecule, along with the symport. So does that make sense? We created a gradient using primary active transport, but then that gradient we can use with this symport to actually drag in the glucose, which doesn't want to come in on its own. So it's still active transport because you're using energy, but the energy is not from ATP directly. The ATP is from, the energy is from a concentration gradient, which was created by ATP before, but it's not using ATP at the site of the pumping right now. Clear as mud.
So are antiports primary active transport?
Not necessarily. In the example I gave here, these are antiports that are also acting as primary active transport, but not necessarily. kind of back to Courtney's thing, it's like there's all sorts of mix and matches. It gets kind of complicated. So like an example that puts all of these together and is an important concept that, again, we're going to be using throughout the semester. This is going to be talking about how does your small intestine actually absorb nutrients into your bloodstream? How do the tubules in the kidney actually process the filtrate and adjust your electrolyte levels and stuff? There's this concept of transepithelial transport, which brings a lot of these concepts into focus and is important for you to understand. I promise you'll have questions about this on your exam in another couple of weeks. This is a very important concept because, again, it's going to be at the core of all sorts of physiological processes, and it also ties in the stuff we've just been talking about. So we're going to start by drawing an epithelium. An epithelium is just a type of tissue that is creating boundaries. So let's think about your intestines. So for instance, so your small intestine. Inside here, this is the lumen. Lumen just means the inner part. That's where all of your food is kind of digesting. That's where the nutrient molecules are finally getting freed up, as well as the poop and stuff that's going to continue out the other side. And then you're going to have some epithelial, maybe I'll do a different color. You'll have some epithelium, your mucosa. There's going to be blood vessels on the other side. And we have to pull, I'm going to use a different color maybe. We have to bring the nutrients across the wall, across this epithelium so we can absorb it into the blood vessel. So this is kind of the picture of what I'm drawing here. And I'm going to now draw it much. So this is, if you probably remember from your class, your anatomy, what kind of epithelium lines the small intestine? I know some of you know this.
Columnar.
It's simple columnar epithelium. Simple columnar epithelium is just a bunch of little, so these are my cells. These are my epithelial cells. This is supposed to be the lumen. And this is where you've got like the little glucoses that are, here's glucoses that are freeing up because the digestive processes. And there's also lots of crap stuff that's going to end up being your poop. On the other side of this epithelium are the capillaries. This is where you are going to be absorbing the nutrients, so you can then carry them and deliver them to all the different parts of your body. So the idea in here, this is my epithelial sheet. So is this making sense? This is the lumen. This is the inside of your intestine. This is on the other side of the wall. This is the inside of your body where the capillaries are. And what we need to do is we need to get this glucose pull it across this wall of cells, and so we can absorb it into our bloodstream. Because anything that stays inside the lumen eventually ends up being your poop, right? So we need to pull over the stuff we want and let the other stuff go. The way it's going to work is going to be a mix of the stuff we've been talking about. So I'm going to draw some parts in here now. This should look familiar. It's the thing I was just talking about. In fact, we're going to couple it with the same thing. Then I'm going to add one more thing that we did not have in the last picture. Okay. All right, so now we have got this whole thing set up. And so what is happening right here? We're burning ATP up, using that energy to pump sodium out of the cell. This is an example of what?
Active transport.
And what kind of active transport?
Primary.
Exactly. This is some primary active transport going on. Right? We're using the power of ATP to pump sodium out of the cell. But now we've created a concentration gradient of sodium by doing this. And so up here, we have glucose that is getting pulled in with the sodium that is trying to come back in. So what do we call this that's happening up here?
Secondary active transport.
Exactly. We've got secondary active transport happening where the glucose is getting pulled in, powered by the concentration gradient of the sodium. And then as the glucose builds up in the cell, and again, glucose is getting taken away by the capillaries. So there's not much glucose here because the glucose keeps getting picked up and taken away. What do we call this? If we have glucose that is going to move out of the cell through a carrier along its concentration gradient, lots of glucose building in the cell, and then it leaves due to its concentration gradient. What's happening here?
Passive transport.
It's passive transport. And what particular flavor of passive transport?
Diffusion.
What's that?
Is it diffusion?
And even a little more.
Facilitated diffusion.
Facilitated diffusion.
Exactly, facilitated diffusion, because we're using a carrier.
Could you explain what makes it facilitated diffusion? I don't totally understand that.
That's what we were talking about just during the quiz, even just by definition. That's what facilitated diffusion means. The word facilitated diffusion is used to describe diffusion that happens using a carrier. So it's just the definition of it. That's what makes it facilitated diffusion. where it's passive transport, things are moving along their concentration gradient, but the only way they make it across the membrane is by use of a carrier. So any questions here? This is critical to understand because it ties in a lot of the concepts we've just been talking about. And plus, like I said, it's going to be at the core of lots of this control of what makes it into the body, what doesn't, maintaining boundaries, pulling things intentionally into the body, like sugar, letting other things move on. We're going to see this in lots of the systems we're going to talk about as we continue through the semester.
For the carrier, is glucose the carrier itself, or what is like?
What is a carrier? This
Yeah.
was. So what is a carrier? Can someone help?
Well, so basically sodium is bringing glucose in.
Right now, but what's the definition of a carrier? Anybody?
It carries the ions.
But
It's a protein.
it's a protein. It's a membrane protein that moves. And remember, this is part. I mean, so obviously, maybe I was not clear in the review at all, because remember I was talking about facilitated diffusion, about carriers, channels versus carriers. Remember this whole idea. A channel is a membrane protein that lets ions across. A carrier is a membrane protein that lets glucose, like sugars and amino acids across. So a carrier, it's a membrane protein. Is that clear?
Yeah.
Yeah, so remember, because all of this is, we've all been riffing off of ultimately talking about the role of membrane proteins like channels and carriers. This like Symport here, it's another membrane protein. It's a bunch of amino acids connected up and folded up in this big complicated thing embedded in the fluid mosaic of the phospholipid bilayer. This little puppy here, this pump here, it's another membrane protein. Bunch of amino acids connected up and folded together, and primary, secondary, tertiary structure to make this big complicated structure that's now embedded in the fluid mosaic of the phospholipid bilayer. Okay, other questions? Yeah, so the carrier is what allows the glucose to cross the membrane. Right? Glucose, large polar molecule, does not want to cross a phospholipid bilayer without help. So it needs help. Oh, the carrier will help. Other questions about epithelial transport? No? Okay.
I actually have a question. So is epithelial transport always going to be like simple columnar epithelial?
Not necessarily.
Oh, okay. This was just the example in the small intestine.
The example in the small intestine.
Okay.
Right. In your like, blood vessels, like capillaries, there's some active moving things, which case you're going across like simple squamous and stuff. Right. So it can be, this is just this particular example I gave, which I thought would be kind of more intuitive, given that most people think about absorbing nutrients from your small intestine. In your kidney tubules, it's going to be transepithelial transport across cuboidal epithelium. All right. If you are confused about anything here and you're just too shy to ask, please go back and make sure everything I'm mentioning here ultimately just makes sense. And it's not like, I think I kind of sort of get it. Make sure it's like, you know, the light goes off. It's like, oh, that's what's going on, right? Because again, this is critical concepts for the whole semester. Okay. So at this point, we are talking about transport proteins, and we're going to now move on to other kinds of membrane proteins that are also just as important and are going to go deep into as well. So I need to clear these drawings. Okay. So far we've talked about transport. What are other jobs that these membrane proteins, again, these membrane, by definition, membrane protein. Here is my cell, my phospholipid bilayer, and I've got all these different proteins embedded in the membrane doing different jobs, part of that fluid mosaic model. So transport, some of them are for facilitating the movement of things in and out of the cell. What else?
Cell signaling.
So signaling. So I'm going to say receptors. So by receptors, those are kind of like the eyes and ears of the cell. These are sitting on here. And again, the things that are going to bind some ligand. You know, this is related. Remember we talked about the ligand-gated channels? Those are channels that have kind of built-in receptors. But we're going to see that they don't necessarily, the receptors are often not connected to a channel. They might just be on their own and set up a chain reaction that we're going to be talking about in more detail. So this could be a receptor for a neurotransmitter. So a nerve cell knows that it's getting talked to. This could be a receptor for a hormone, which is a signaling molecule. So the body knows that insulin just got released. Or it might be like your taste bud. Receptor on your taste bud for sugar molecules is how you detect like sweetness in your food. Receptor, so this is just, it's basically allowing the cell to recognize specific molecules outside. And again, ligand, by definition, ligand is just a signaling molecule. And again, this we say that the ligand binds the receptor. It makes this kind of temporary connection. It's kind of, you know, activates something and then it lets go. We are going to look in much more detail again what binding actually means, what happens to the receptor when binding happens that makes the magic happen. But first, I want to stay a little more superficial. So basic idea makes sense about what a receptor is, what a ligand is. Okay, so now we can talk a little more detail about this. Actually, let me do. Come on. So endogenous ligands. Endogenous ligand just means ligands that your body is normally making to do its stuff, like serotonin or insulin or any of the other myriad signaling molecules that your body uses to communicate between cells. However, there's the idea of the exogenous ligand. So exogenous ligand. So, and this is just from the word endo, within, generated, created within ligand. These are just like serotonin or dopamine or adrenaline or whatever. Exogenous, generated without, comes from outside the body but mimics the endogenous ligand close enough to bind the receptor. So again, we have some cell, we have some receptor, and you have some ligand that normally fits in there. It might be that there is some other thing which from outside that has a shape close enough that it can also get in there and bind. This is how a lot of drugs work, right? This is how, this is why you get high. If you're smoking pot, there are endogenous ligands, neurotransmitters, the endogenous cannabinoids like anandamide that bind the cannabinoid receptors. THC, on the other hand, has a close enough look that it gets in there and also binds and has an effect at the receptor. Except the THC is an exogenous ligand. You got it from like burning some plant matter and inhaling it, and it got into your body that way. It's not created in your body. It came from outside your body, but it has a shape and chemical properties close enough that it can bind the receptor and have an effect. So does that make sense? So let's take a look at this a little deeper. So exogenous ligands, things that are binding receptors but are not actually the original thing. There's two different classes. There are agonists, bind and activate the receptor. And antagonists, bind and block the receptor. Do not activate it. So, examples, agonists. Like if you're having an asthma attack, you have a little albuterol inhaler. albuterol is a drug, an exogenous ligand that binds and activates adrenaline receptors. And those adrenaline receptors then dilate your bronchial passages so you can breathe easier. So like albuterol inhalers, those are an example of a agonist for adrenaline receptors. There are lots of like morphine or heroin or Vicodin, any of the opiates. Those are agonists for the opioid receptors. They bind the opioid receptors and they activate these pain blocking pathways. Right? So an exogenous ligand that binds and activates the receptor, like morphine or like albuterol, those are agonists. An antagonist binds the receptor, but it doesn't activate it. But because it's binding, it's keeping other things from actually getting in there. So an example of antagonists are like beta blockers, things people take to keep their blood pressure down. Those are actually adrenaline antagonists, things that will keep adrenaline from getting in the receptor and keeps your heart rate down, keeps your blood pressure down. Right? So an antagonist is something that will block the receptor. So does that make sense? Some things will bind and activate the receptor. Some things are useful drugs if they bind and block the receptor.
I'm sorry, what was your example of an antagonist?
I was saying like beta blockers.
Oh, I'm sorry, I meant agonist. Sorry. No, I got the beta blocker. What was the agonist?
Oh, albuterol.
Albuterol.
Is the stuff that's in like an asthma inhaler.
Yeah, okay, thank you.
Would methadone also be an agonist?
Yeah, absolutely. All of those opiates that will turn on that system. And in a few moments, we'll talk about an opioid antagonist, which would be naloxone, like Narcan, which does the opposite. It gets in there, but then doesn't activate it. There are things in between. There are things called partial agonists, which will turn it on, but not as strongly as a full-blown agonist. So there are things in between. But for right now, just think about agonists and antagonists. And then the other concept that we need to think about is affinity. Affinity works in parallel with these. This is how tightly the ligand binds to the receptor. So let's do an example here. I need to, so high, low affinity, speaking about opiates. If you want to give somebody enough morphine so they have pain relief, you have to give them maybe like 30 mgs of morphine. You'll be able to like kind of exist in your body, even if you've got like some broken arm or whatever. Fentanyl, which is another common opiate agonist that they use in a clinical setting, you only need 100 micrograms, right? 100 micrograms, 0.1 milligram. So how is it that you only need 100 mics of this, but 30 mgs of this? If you think about, here's the receptor. If something comes in and activates it, then goes out, then comes back in and goes back out. It's got a certain amount of activity there. If you have very high affinity, you get in there and you just hold on and you keep activating it. So you don't need as much of the molecule floating around if you have high affinity, right? Because once it gets in there, it stays. Things like psychedelics. If you want to like trip with mescaline, you're going to need like at least 200 milligrams or something. Whereas you can have a very strong trip with only, again, about 100 micrograms of LSD. So, you know, like a 300, what was it? Yeah. 10, yeah, over 300-fold amount of, you know, kind of how efficient this molecule is in activating the pathways. So high affinity, things grab onto the receptor and keep activating it. So you don't need as much around. Low affinity, it does grab, but then it lets go, and then it grabs and it lets go. And if there are things with higher affinity, they're going to get in there and they'll outcompete it. So we should talk about Narcan. This is an important. This is a high affinity opioid antagonists. This is something that is in most like people's like emergency response kits now, and it will save somebody's life if they have an opiate overdose. The basic idea here is, let's say somebody just took way too much, some opiate, whatever it is. It could be morphine or heroin or OxyContin or whatever. So they have their receptors, they have, let's say it's little heroin molecules zipping around and getting in the receptor. And one of the problems, if you have too much of an opioid, in addition to making you floaty and feel good, it suppresses your breathing and you're gonna like suffocate and die. So what you need to do is somehow stop that effect of that overdose. So Narcan comes in, high affinity, it gets in the receptor. Once it's in there, it has a much higher affinity than these other heroin or OxyContins or whatever are floating around. So it stays in there, and these dudes are just kind of waiting in the wings, and they're just floating in your bloodstream, but they're not actually binding the receptor, making you have the effect. But Narcan is an antagonist, which means even though it's grabbed on there super tightly, it's not activating the receptor. So you're pretty much instantly no longer under the influence. I've never been there when someone's got the shot, but people who have told me that people are often like super pissed. It's like they were just floating away, and then all of a sudden it's like, what the heck's going on? What did you just do to me? I was feeling really nice, and now I'm here, and because pretty much they're like instantly back. You know, you just need to make sure you keep the levels of the naloxone high enough in their body until the body has a chance to clear out using its normal methods the other opiates. So, you know, by the time the Narcan's gone and this is gone, then they're kind of back to normal. But so this is showing high affinity. It grabs on and blocks the overdosed opiate from getting into the receptor. And it's an antagonist, meaning that it's blocking but not activating. So does that make sense?
Can I ask a quick question?
Sure.
So just to like make things more clear, so something that has low affinity, like the heroin, like how you drew more of those, do they like bind and then kind of like detach and then another one binds, and that's why they have low affinity, whereas the other one binds?
Exactly. Yeah. So, you know,
Okay.
I'm trying to think of a, I don't know, like maybe like in a social thing, like, there's somebody that just likes to give someone like a quick little hug and move on. It's like, oh, so nice to see you. Nice to see you. Nice to see you. And nice to see you. Whereas the other person's like, oh, it's so nice to see you. I just want to just feel what it's like, it's like, just, it holds and it doesn't freaking let go. As opposed to it holds, but then it goes off to what's, lets go and then maybe comes back. So it's kind of like that.
Okay, thank you.
Sure.
And then you said something high affinity obviously will like overpower the low affinity. So if you bring in something high affinity, it cancels out whatever the low affinity.
It's not, I mean, overpower is, I'd say it kind of just hogs all of the space. Like if, it's like let's say there's one receptor, if there's some low affinity thing, it comes in, it comes out, it comes in, it comes out, so it's having its action, but it's doing it in this kind of coming in, it detaches because low affinity means just all the wiggle makes it unbind, then it binds, and then it unbinds. But if you have some high affinity thing that comes in, it's not letting go. This thing doesn't have the opportunity to just bind and unbind and bind and unbind, because every time it comes in to bind, this thing's still hogging the receptor because it holds on and isn't letting go.
So does the low affinity, does it just like, where does it end up going? Does it just like dissolve, or?
Your body has different ways to clear stuff out of your bloodstream. There's also your liver, your kidneys. Your liver has all sorts of enzymes that break things down, detoxify things. Your kidney tubules are constantly filtering and removing things from your plasma. So those are the normal processes. Like, right, if you take a shot of tequila, you're gonna be like buzzed for a little bit, and then you don't stay drunk the rest of your life. Like after like a half an hour, you're probably ready for your next shot, right? It's because you've got enzymes breaking down the alcohol in your bloodstream and deactivating things and eventually excreting the waste products and stuff. So yeah, your body is constantly clearing stuff. Like when we'll talk about this more. Whenever you talk about some drug, we talk about, like, if you look at the plasma concentration over time, whether it's caffeine because you just drank some coffee, or whether it's aspirin because you just took some aspirin, or it's constantly getting, and we talk about the half-life. The half-life is how much time until half of it is cleared out of your bloodstream. And then it's like an exponential thing. One more half-life, half of that is cleared from your bloodstream. Another half-life, half of that is cleared from the bloodstream. So whenever we talk about drugs, we always talk about the half-life, which is how long before about half of it has been cleared out through all these mechanisms that your body has to kind of bring you back to homeostasis after you have, added something to the mix. All right, so we talked about transport. We talked a bunch about that. This should start feeling like, oh yeah, channels, carriers, symports, antiports, active transport, passive transport, transepithelial transport, all this kind of stuff got kind of tied into there. Receptors, receptors. Again, kind of the eyes and the ears of the cells, how they can detect what's going on outside of them. It's going to be important both for communication between cells, like cells can release neurotransmitters signaling molecules to talk to each other. It's going to be important for cells just to have a sense of their environment, like what's out there, how do I need to respond to it? And again, we talked about the idea of ligands binding receptors and endogenous versus exogenous ligands, and exogenous ligands can be antagonists or agonists and can have high affinity or low affinity. And then the rest we're going to go through kind of quicker. These are the ones we're going to be spending a lot of time on as the semester goes on. The other ones we will be meeting along the way, but they don't have as deep profile, so to speak. So what are other things that membrane proteins are doing?
Are there some that anchor themselves to other cells? Like,
So cell to cell attachment. Exactly. So, you are made out of like 10 trillion cells, and those cells are stuck together. You're not just like a big slurry of cells. They're all stuck together and make you. And if you've been in anatomy, you've looked at this in much more detail. I will just really briefly review this. So remember in transepithelial transport, we have our cells, and we are pumping glucose across and everything. There was the lumen. There's like our blood vessel in here. So we want to make sure that the sugar comes across and we get into our bloodstream. But there's also stuff in the lumen here. It's supposed to be like a little turd. It's just the stuff that's going to become your poop, right? So you only want stuff to move across the epithelium that you are intentionally moving using the transepithelial transport, which means you need to have some kind of seal between the cells so things can't just wiggle from one side to the other, just kind of sneaking around the backside there. So these little junctions, these are called tight junctions. They're a lot like Ziplocs. Think about a Ziploc. A Ziploc, you have two sides, but then they interleave in such a way that it becomes even waterproof. But then you can pull them apart. It's, the two pieces of plastic are still two pieces of plastic. But the Ziploc junction allows those two things to really have a super tight seal. So that's what like tight junctions are like. So any like random thing that's trying to get across cannot make it across. The only things that are making it from one side to the other are the things that we are intentionally moving. So that's tight junctions. You should, so another kind of junction are desmosomes. We're not going to really talk about them all that much, but you should know they exist. Especially like cells where there's a lot of physical force yanking on them, and you want to make sure they don't pull apart, like the skin cells. You don't want to like scratch your skin and have your skin just shred up. You want those cells to really hold on. Your heart muscle is pulling really hard to push your blood through your body. You don't want the walls of your heart, tearing apart under the stress and the pressure. So what you have are these desmosomes. They're like little rivets, and there's all these intermediate fibers which act like kind of like the cables that basically anchor the cells together so they can't pull apart. So I usually think of them as like rivets and cables. But basically the desmosomes are about kind of structural integrity, just keeping the cells from pulling apart under force, under tension. And then the last one I'll mention are the gap junctions. So here's the cells. The gap junctions are actually like little tunnels. They're called connexons. They let stuff drift between the cells. So they actually connect the cells. For the purposes of our class, we just need to remember that gap junctions let things move between cells, including ions. And because ions are the electrical current carriers, if we have gap junctions, it's like kind of electrically connecting the cells. So this electrically connects the cells. This will be important as we continue on in the semester. We'll see particularly like cardiac muscle, smooth muscle cells are connected with gap junctions. If one gets electrically excited, all its neighbors get excited. In the nervous system, most nerves talk through neurotransmitters and receptors, but there are some that are talking with electrical synapses where you actually have gap junctions. So it's important to know that you can electrically connect cells with gap junctions. So these are the three main kinds of cell-to-cell attachment. Tight junctions that just make a Ziploc seal. Again, super important in things like transepithelial transport to make sure that the two sides are separated from each other. Desmosomes, rivets and cables, we won't talk that much about. Gap junctions we will see from time to time as ways to electrically connect cells. All right, so we did cell-to-cell attachment. What other things could we have? Membrane proteins, what do they do for you, for your cells?
Would endocytosis and exocytosis fall under the transport category?
Yeah, I would call that part of transport, but yeah, the clathrin is indeed a membrane protein and kind of fundamentally different from the channels and carriers. So I could see you thinking about them as a separate thing, but I probably call them transport.
Okay, thanks.
So other things, cell identification. You know, you've got 10 trillion cells that make you, but there are all sorts of other cells that aren't you that are trying to get in or try to set up camp, make you sick, all these pathogens and, you know, so all of your cells have specific markers on them that say they are particularly belonging to your body. We'll talk about this in much more detail when we get to the immune system and talk about like major histocompatibility complex. But you know, this is why even if you needed a kidney and I was happy to give you my kidney, the chances are it wouldn't work. I would give you my kidney and your body would say, these cells don't have the right ID marker, kill it. Right? So, you know, cell versus host disease, or graft disease, if you're trying to, graft versus host disease, that's what I'm trying to say, graft versus host disease. If you're trying to get a organ transplant, it usually doesn't work very well unless the two people are really closely related, because all of your cells have a specific marker that say they're part of your body. And if your immune system does not see, your papers are out of order, you're dead. So that's kind of part of keeping your body safe, is making sure all your cells have the appropriate ID markers.
If the organ is closely related, will the body reject it still, or?
It won't reject it as fiercely. You still need immunosuppressant drugs for the rest of your life, but at least it'll work. Whereas if it's really quite a different organ, then no matter how much immunosuppressant you're trying to do, it's going to fail. All right, cell ID. What other things do proteins do in the membrane? I'll just help you out. Right, your cells are not just bloopy blops, right? Some cells look like this. Some cells look like this. Your blood cells look like biconcave discs. Some cells even move around, like, so all of that means that you have the cytoskeleton attached and holding on and keeping its shape. And when a cell does endocytosis or endocytosis, you're pulling in or pushing out. So there's all sorts of cytoskeletal attachment points that are membrane proteins. Another thing that happens, membrane proteins can be enzymes. If there is some reaction that you want to occur right near the membrane, then it makes sense to anchor the enzyme on the membrane. So, for instance, we were talking about the transepithelial transport again. The final steps of breaking down your nutrients, where you're going to end up with the glucoses. When you've got the glucose right near here, you want that to happen right near the membrane, because you want the final products of digestion to end up right near the membrane where they're going to get absorbed. So you're going to have we'll meet them in the digestive system in like 3 months from now. The brush border enzymes are all anchored right along the membrane itself, the enzymes that do the final steps of digestion. When we get into cell signaling next class, there's going to be enzymes that are specific to help with the receptors do their job. And those are going to be anchored right near the receptors in the membrane. So we'll see this through the semester, that enzymes will sometimes be anchored right in the membrane if it makes sense to have the reaction happening right near the membrane. So transport, receptors, cell-to-cell attachment, cell ID, cytoskeletal attachment. I wonder if I've left anything out here. Hopefully not. Yeah, I'm going to just leave it there.